Welcome everyone. On behalf of Pre-Science in Silico, I welcome you to the Fusion Talk. A few words about Pre-Science in Silico. At Pre-Science in Silico, we team up with both industry and academia to tackle challenges in drug discovery and material science. The idea for this talk series came from our wish to create a space where experts from different fields can share their valuable insights. We are proud to be the first Indian company to create our own drug discovery software with advanced AI features. Pre-Science was founded by academicians who wanted to make AI-powered discoveries in drugs and materials accessible to everyone, aiming to redefine the landscape of scientific innovation. The title of today's Fusion Talk is Consequences of Machine Learning on Structural Protein Biology. Let me introduce the speaker for today's talk. Dr. Ratul Chowdhury is an assistant professor of chemical and biological engineering at Iowa State University. Their research group, ChowdhuryLab, also known as Structural Protein Biology and Engineering Lab, focuses on developing and implementing novel machine learning strategies to predict protein structures solely from their amino acid sequences. Their work spans various applications, including drug discovery, biomaterial design, room temperature breakdown of complex molecules, and precise molecular separations. Prior to joining Iowa State University, Dr. Chowdhury earned his PhD in chemical engineering from Penn State University in 2019 under the supervision of Professor Costas D. Maranas. During his doctoral studies, he specialized in optimizing the tools for the redesign of channel proteins for separation, enzymes for altered substrate and cofactor specificity, and de novo design of non-immunogenic antibody variable fragments against disease-causing proteins. Following his PhD, Dr. Chowdhury served as a postdoctoral fellow in the computational structural biology unit at Harvard Medical School, working in the lab of Peter Sorger in the Department of Systems Biology. I hope I mentioned it correctly.
Yep.
Thanks. Here, he focused on devising machine learning strategies to accurately predict the structures of voltage-gated ion channels and kinases. Additionally, he conducted research on docking a library of drugs to identify non-opioid drug-binding modalities with implications in pain and cancer research. As you can see, we have an expert in machine learning, and he has been working in machine learning for a significant amount of time. So we are delighted to have you here, Professor Chowdhury. And a note for participants, please feed in your questions in the chat box. We'll address these questions at the end of the talk. Now I request Professor Chowdhury to start the talk. Thank you.
Right. Thank you, Anupam. Thanks for the great introduction, and this is my pleasure to be here. I will sort of, go over the overall journey. Sort of, it's machine learning did not sort of, pop up from day one. So I will sort of give you a brief journey of how things ultimately became machine learning driven. While we do machine learning at the front end, we try to make sure that there is relevant amount of bio awareness in whatever we are doing, so that it's not just cranking away computational power and throwing data at it and seeing what comes out of it. So that is where sort of what is our unique positioning is. And before I get started, a brief thing about Iowa. So most of you might have heard this. So Iowa, Ohio, and Idaho. I get a lot of confusing requests that are you at Ohio, are you at Iowa, or are you at Idaho? But these are three different places. So I'm at Iowa, which is right at the center of the country. It's called the Midwest of the U.S. And this is what this is a pretty flat plain, which is a pretty agricultural state, and so this is very, very big on cycling. So we have the largest cycling track in the whole of the USA. We have something around 500 miles of cycling competition every year. It's called the RAGBRAI competition. This is what Iowa is known for. The track actually goes through the campus, so it's like an event in summer. And this is how the campus looks like during winter. So we have a pretty strong winter. And this is pretty much around the end of the winter at this time of the year. Okay, that's so much for Iowa. And then now let's getting started. I think I don't need to motivate this crowd about what proteins are, but again, for a general audience or for anybody who's listening to it separately outside this live audience. So proteins are unidirectional polymers. They are made up of amino acids, which you can imagine as like beads in a stringed necklace. And these beads could be of 20 different types. They have different charges, sizes, shapes, etc. And if you can string them together based on the individual charge interactions and stuff, they will fold into specific three-dimensional architectures. And that's what is important, because if you look at the left top, you'll see that a protein does not really stay like a stringy polymer, like a beaded necklace. It actually folds into a very precise shape. And on the left bottom, you can see that even if you make a very small change on the left side, because it's one connected chain, it's a single chain, since I'm thinking that most of you are computationally savvy in this crowd, so I would like to refer to proteins as a one-dimensional curve in a three-dimensional space. So if you make a small change on the left hand, it'll sort of propagate downstream to the right-hand side and can induce a long-range structural change. And also on the right side, you can all see that this is a very popular protein by now. We've seen this. This has made it to the news. So this is the SARS-CoV-2 spike protein. The interesting point to be made here is that you don't really need to make changes onto a protein to bring about a structural change. So these three proteins that you can see, these are all identical proteins, but the blue one undergoes a structural conformation when brought into the vicinity of the red and yellow one bound together. So the protein structure undergoes changes for a lot of different reasons. And we can only understand, like, whatever we can see, and there could be many other reasons that could be at the back end. So our lab and the general focus of our research is sort of divided into three different themes. In the left side, you can see the first theme is basically take a protein, make small changes onto it, make sure that the structure remains intact, and endow it with local new properties. The second theme is basically take a protein, make drastic number of changes onto it without disrupting its structural integrity. At the same time, make it to do something very, very new. And then there is a third piece, which I don't think I'll have enough time to go into today, but happy to chat about it later, is that we take inspiration from protein structures and what they can do and design synthetic architectures with similar properties like proteins. So the first piece is that take a protein and make small changes onto it. So this is the paradigm of enzyme engineering. This is the one paradigm I'm going to talk about today. So we have a protein structure prediction tool, protein design tool called iPro plus minus. The thing with enzyme engineering is that there are at least a dozen tools out there which can perform enzyme engineering. But why again come up with one more tool? The reason for this is that most of the enzyme engineering tools cannot predict new deletions or insertions. So if you look at the bar graph at the bottom, I'm showing you the family of the TEM beta lactamase protein. So there are 34 members of this TEM beta lactamase, which means the 34 species have TEM beta lactamases, which are exactly 275 amino acids long. But if you just start with that protein and perform only substitutions, you're going to keep the length fixed, and you're going to miss 62% of the sequence space, which basically is a loss when you're trying to do protein design. You don't want to miss out on sequence space. So, and if you look at the top picture, you'll see that the sequence alignment, performing a simple sequence alignment, tells you that there are prevalence of dashes. Dashes means that the length of all these family proteins are not the same, which basically refers to the fact that nature uses the length of the protein as well as the sequence itself as a design variable. So for a computing crowd, I can even understand what a design variable is. So we said that we will allow this as a design variable. We'll help it to identify favorable insertions and deletions. So this tool was made in the pre-machine learning era where we couldn't predict using things like AlphaFold, ESMFold, and so forth. So we had to basically say that where are these insertions? How do we find where do we make these insertions and where do we make these deletions? So as a basic default option, what the tool allows is that, if you perform a sequence alignment, if you see that there is at least one dash in the starting sequence that you're trying to engineer, that means there is at least one family member which allows one extra amino acid there. So that gives you some sort of an idea of probability of making an insertion there. But in the same way, if any of the family members has a dash in a certain sequence where an alphabet exists in the starting sequence, that's a possibility for making a deletion. So we use basic probabilities from that to make insertion and deletion. Again, this is since there's a pre-structure prediction era, we had to figure out how do we make an insertion deletion. So we took inspiration from the inverse kinematic problem, which is largely used for robotics, how a robotic arm optimizes its elbows to minimize the amount of energy consumed to grab onto a stationary target. That's what we did for performing an insertion or deletion. So insertion and deletion is slightly different from a substitution because you have to induce a polypeptide break and then you would have to insert your amino acid of interest, and then or delete the amino acid of interest, and then join back the free ends. So the fact that you have to join back the free ends, we need an inverse kinematic problem which can minimize the stress between the hands. And while we were doing it, we made sure that all the side chain angles were preserved in terms of the Ramachandran plot was not violated. So once you look at it from visual perspective, this is what it looks like. You have a insertion. We insert a glycine, which has the option to become something else later on, depending on if it's a favorable move or not. And on the other hand, for deletion, it's much more simpler. You delete the amino acid of interest. So this got plugged into an existing tool, which is called iPro, Iterative Protein Redesign and Optimization Toolset. And in this case, now you can perform insertions and deletions. It's an automated tool. It decides for itself after making a glycine insertion in the stage 2 at the rotamer repacking stage, whether it is favorable for it to choose a different amino acid to favor the binding of the substrate or leaving of the product. So, and again, later on, we also have an option of checking whether to make a deletion, and everything else around stage 4 and stage 5 of this tool goes to this Metropolis criterion, where it actually checks whether it's a good move to make this insertion or deletion. And this can add this on top of substitutions, so which is a good combinatoric gain. So ultimately, after several cycles of this design, we'll get a bunch of engineered enzymes. So we've published extensively using this tool, and which have for engineering enzymes, largely thioesterases, coumarin-CoA ligases, for lignin valorization, for biofuel precursor design, and also design of non-ribosomal peptide synthesis. And these have been validated experimentally, and we are currently using some improved version of this tool, which has ML-based components, and also which can learn from the negative data sets. So if you perform some experiments, let's say 30% of the experiments or like 40% of the experiments don't turn out to be right, the proteins do not express, do not fold, or do not purify, there is some issue downstream. So all of those data does not stay in the lab notebook. They actually come back to our software and say and informs the software that this was a bad mutation for whatever reason, based on this particular experimental workflow that we have, this does not work. So avoid this kind of a mutation and try to come up with things that are not similar to that. Okay, so I'll move, sort of switch gears and move to the next part of the story, where we take a protein and sort of make a lot of engineering on it, make a lot of amino acid mutations on it. So this paradigm is for engineering pore proteins. So this part is called pore designer. So pore proteins or porins or channel proteins, as you might call them. So they are present. These are transmembrane proteins which can connect help an organism connect with its exterior surrounding. For the case of a bacteria or single cellular organisms, this is very important because it helps them retain their osmotic balance and help transport water, transport sugar, salts, amino acids, and a lot of things. So we said that, okay, so we'll take these out of the living world. We'll take these pore proteins outside, sort of, rupture open the bacterial cell wall and purify and extract these proteins out and constitute them back in flat sheet membranes and use them as separation devices. So one of the primary test cases or benchmarking we had to do to check whether it's actually possible was to see if it can be used for just permeation of only water. Anything bigger than water should be rejected through this pore, and anything smaller than water, anything bigger than water should be rejected. So water should only go through. So this is again, so nature already has a protein that can do something like this. So this is called aquaporin. So aquaporin is a pore protein which is alpha helical and allows only a single water to go through. This is the mechanism through which it lets water go through. It actually does not let protons go through. It only lets water go through. So, and Peter Sorger won the Nobel Prize for describing the mechanism of proton rejection sometime around that time, 2008 or thing, I think. So we were saying that if nature already has a protein like it, then what's the problem? So the problem here is that if you try to put this alpha helical protein in a flat sheet membrane, during the sheer stress of the operation of a filtration process, this alpha helical pores actually open up and they let bigger molecules go through. So we said that we'll start with a much more rigid, much more structurally stable protein, like a beta barrel protein instead of a alpha helical protein. The problem with beta barrel proteins is that their pore sizes are actually much, much bigger, roughly 2.5 times the pore size. So we said that we have to marry the two concepts. We have to marry the selective internal pore geometry of a helical protein and graft that whole thing inside the pore protein, at least conceptually. So my experimental collaborator said that we can make this OMPF. If you can design your OMPFs, the smallest black dots you can see in this picture on the left, those are individual proteins. So there's roughly 1 million proteins per square millimeter of real estate. So he was like, we can lay these out as flat sheet membranes using our layer by layer technique, and we can cast them on a porous support, and thus we have like a functional filtration device. So the objective was pretty clear for us. Okay, we have a big pore size protein, which is probably stable to mutations. We have to plug the pore enough that only one water goes through. So we looked at the pore constriction, the picture on the right, and we said that we will jam it up with tryptophans. So tryptophans are hydrophobic, and tryptophans can actually clog the pore enough and lead to the smallest pore size that we can get. And that will tell us what's the lowest limit of pore size we can get to. And we had to test that out. So the problem with that was that when you jam a lot of tryptophans along the pore perimeter, consecutive tryptophans actually start clashing against each other, and one tryptophan would face out of the screen that you're looking at, and one tryptophan would be facing inside into the screen. And so ultimately the pore size would become much, much bigger than what you expect. So at this point, we said that, okay, this is actually a perfect optimization problem because we have to place hydrophobic groups. So if this is not clear, why do we want to place hydrophobic groups here? That's because there is water that is going into these pores because of osmotic pressure. So water is anyway coming inside because of the salt gradient. So now question is, if the water is going inside the pore, you don't want it to stick around in the pore. You want it to come out of the other side of the pore ASAP. So that's why you want the pore walls to be extremely hydrophobic, much like carbon nanotubes. So we said that, okay, we will now place small and big amino acid side chains alternately at the pore periphery. But how many alternate such combinations can be made from these things? So that's why we cast this as an optimization problem. This is largely an integer optimization problem. So this is what the optimization problem looks like. I can come back to this slide if there is somebody who's interested in knowing what each and every step does. But ultimately, when we design this optimization program and sort of run it through our molecular simulation platform, what we see is that we were able to get some designs. We were able to get some designs that we could experimentally validate. And we found an extremely hydrophobic design, which allows, which is not only hydrophobic, but also has a pore size small enough that does not let anything go through, like NaCl, glycine, glucose, sucrose, PEG 600, all in increasing size. On all of these things are rejected, whereas the wild type pore lets a lot of things to go through. And then what we did is that we said that, okay, we started with a 10 angstrom pore. We were able to reduce it to a much smaller pore size. So how about we get all the intermediate designs that are accessible through this pore design platform? So the key point to take away from here is that this pore will not be used for desalination. This pore will not be used for purification of water. And I can tell the reason is that we did a market analysis. If we have to purify water using that pore, 1 gallon of water, which is 3.78 liters of water, would be something of the order of $40. And we can sort of buy 1 gallon of water for $3 right now. So the thing is, why even do this? So the utility of this is that you can separate, which are two similarly sized drugs, where one is a drug and the other is an impurity, where each of these drugs are actually much more expensive than water. So this is a platform technology which can be used for precise separations with therapeutic pharmaceuticals and a lot of protein separations in the mindset. So we recently performed a review of the last 40 years of pore designing efforts and where these pores have been engineered. So if you take a pore protein, which part of the pore do you need to engineer to perform what kind of task? The task being separation, sensing, and sequencing. So we did a comprehensive review across the patent literature, across the publication literature, so that covers industry as well, academia alike. And we lay down some ground rules that explain why these design strategies work. So then this is the part that I'm not going to go into a lot. So this is the part where we say that we take inspiration from protein pores and then also design synthetic architectures. So the primary goal for this is that if we design a protein pore, only 20% of the whole real estate, whole circle is actually the pore. So 80% of the area on the membrane is occupied by the protein. That's pretty much a loss because you can increase the efficiency of separation by introducing more number of pores if the pore itself is small in bulk. So that's why we worked with organic cages and supramolecular channels, and we worked on a couple of different types of materials and demonstrated the similar kind of performance using these type of materials. So then I'm moving to basically the third piece of the work, which is sort of motivates, right? So around this time when I was sort of finishing up my PhD, the world was largely moving towards data-driven sciences, and I had joined a lab which was interested in performing, like making a tool for structured prediction. And right around the time AlphaFold came out, and we were part of this review process, and we had some ideas about, we were one of the groups that reviewed this article before it came out. So we also participated in the Cast 14 competition. And what we saw is that AlphaFold sort of beat everybody, also beat their own prior record. They actually won it in the previous year as well, but the performance of the quality of prediction was close to 60%, and they hit roughly 90% in this year, in 2020. So that was phenomenal. So we know exactly why this works. So the reason why it works is you have a protein sequence, you perform a multiple sequence alignment, you look at the conservation of different alphabets across different columns. So if M occurs in the first position, how many times does S occur in the sixth position? So you ask these questions and get statistics from it and map those statistics onto the real physical 3D space distance between these alphabets. If MAFHK is present in 3D space, how far is M from A, how far is M from F, and how far is M from H? You map that 3D information, distance information, onto the statistics. And the question is, why would that even work? Why would statistics from sequence be mappable onto the structure space? The reason is pretty simple. The reason is simple because if you imagine that this is a long protein, and the left-hand side, the N terminus of the protein is this red domain, the C terminus of this protein has a green domain. And imagine that this protein is like a U-shaped protein where the green domain is actually sitting on top of the red domain. If that's the case, the reason for that is there is, let's say, a salt bridge, there is, let's say, a polar interaction, and then there is like a hydrophobic packing. So there are three forces that sort of Velcro one domain on top of each other, resulting like a U-shaped structure. So if this U-shaped structure has to be conserved, for example, this is a protein X in let's say humans. So the same protein X in chimpanzees or ants or E. coli will have the same U structure. For it to retain that same U structure, it has to always maintain the contacts. So if E changes, if the alphabet E changes from humans to, let's say, chimpanzees, the E has to be followed with something that can make a complementary interaction with right on top of E. If the R becomes a D over here, then this E actually has to become a K or a R. So that's to retain the 3D structure. So there is some consequence from structure to sequence. So this is what AlphaFold extracts and leverages on. And it depends on how many organisms it can find as homologs to predict a high quality structure. So that brings it to its caveat. The caveat is in figure 5a. If you read the AlphaFold paper carefully, you must have seen this, is that this is the number of sequences that can be aligned to your target sequence. So this is close to 30. And on the y-axis, you see the quality of prediction. You see that the quality of the prediction takes a dip when you have very less sequences to align against. So that means that it totally or largely relies on the knowledge of known protein sequences, which are similar to your target protein sequence. So this sort of, we did a full analysis on how much computation power or sequence information is processed by all the tools that existed in the market at that point, and what is the prediction accuracy of these tools, and what kind of operations they do to get to this. So at the left top over here is this real biological process of protein folding. That does not rely on a sequence alignment. A protein folding in E. coli does not care about how the same protein might be folding inside yeast. So it does not care about a multiple sequence alignment. So we said that even if at the cost of accuracy, we should come up with a tool that sort of alludes to this process rather than the process of understanding from an MSA. So the reason for that was that proteins do fold as single sequences. So for that, we had to do two things. The first thing is that we had to capture the structure. And the second thing is that we had to capture the sequence. And then we had to map these two mathematical objects. The first thing is to capture the structure. To capture the structure, we describe the protein structure through something called Frenes-Rayleigh transformations. This is basic Eulerian geometry, which says that if you have to travel from one point in Cartesian coordinates 3D space to another point, all I need to know is the two unique angles that can get me from here to there. And again, a set of two unique angles that can get me from here to there. So that's what we recorded. We recorded the whole protein structure as a series of pairs of angles. And that was our description of the protein structure. And then basically we had to map the sequence. So for the sequence, we use something called a transformer model, which is also now called a language model. We call it amino BERT. For a general purpose audience, I'm trying to say that, okay, what it means. So we refer to each protein sequence as a sentence in, let's say, any language. Okay, so for example, if the language happens to be English, let's say there are two sentences: I am from India and he is from India. These two sentences, all of you in the crowd know English, so you know that the verb form that is used exactly here is entirely dictated by what comes before it. So I cannot say that if I say I is from India, all of you will know that that's wrong. There is no reason why you accept that, but you accept it because you've sort of memorized the grammar. There is no other reason why that has to be correct. So exactly what we are saying is that if you give somebody a book which has, let's say, 250 million sentences in English, at some point, if they're able to memorize all the sentences, they will be able to see through the pattern and figure out what is the grammar. So that's exactly what we did using a computer. And instead of giving a dictionary of sentences, we gave them a dictionary of protein sequences, which is the UniProt database. And then we asked the question, how do we challenge its learning? We said that, okay, we'll give you a protein that you've seen before, but let me blank out some alphabets and let you guess the alphabets based on the knowledge of what comes before that alphabet and what comes after that alphabet. So this was performed. Then we went to NCBI conserved domain database and blanked out certain domains of proteins and said that, well, can you predict the whole domain sequence just from what comes before the domain and what comes after the domain? So that test was also performed. So all of these things again work. One final attempt to sort of explain why this works is because if you follow the left sentence vertically, the cat drank the milk because it was hungry. The word it actually qualifies the cat if you use the word hungry. But if you want to use the word sweet, the cat drank the milk because it was sweet, then that same it actually qualifies the milk, not the cat anymore. So there is some inherent dependency between the words cat, milk, it, hungry, and sweet. So there is some positional dependence, and then there is some choice of a word that is also depending the thing. So that's the same thing as saying which amino acids at what positions interact with each other. So that's exactly what we're trying to get at. And our tool, we released our tool, which sort of got published in Nature Biotech, and we also got covered along the lines to discuss about it for about some aspects of it. It is faster. It is not necessarily the most accurate when you compare to AlphaFold, but there are certain classes of proteins where it outperforms like some performance of AlphaFold. So again, for a general machine learning audience, so one needs to know how to use machine learning tools rather than just be excited about it. That's basically what my stance is as an engineer here, is that, so for example, if you do this thought experiment, if you take a multiple sequence alignment, you can feed it to the AlphaFold pipeline. They have CollabFold now. You don't even need to download AlphaFold. You'll get a structure. But if you're able to feed the flipped multiple sequence alignment, the flipped multiple sequence alignment has no meaning. These are all flipped proteins and does not probably mean real proteins. You are going to get a flipped protein as an output because it learns from geometric objects and tries to map it onto like the multiple sequence alignment objects. So one needs to know what they're doing, because if you imagine, if you make this sequence very, 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 very short, if you just are looking at 5 amino acids. If you're just looking at only 5 amino acids, the flipped sequence might actually truly be a real sequence. So in which case you have no idea what you just predicted. Did you predict the right sequence or you predicted the flipped sequence? So the bioawareness angle is really, really important. And there are proteins for which you actually don't have any MSAs. If I take the protein from extremophiles, if I take the protein from the bacteria that grows at 100 degrees Celsius in a hot spring and grows under a condition of 1 pH, so that protein has got no obligation to repair any homology with any protein that's out there. So what will you do with AlphaFold? So AlphaFold is not going to give you any multiple sequence alignment. If that protein is not a part of its training set, it's going to give you a bunch of loops, or it is going to make some grave assumptions. So that's where we sort of come in as a picture, as a solution, that if you learn from single sequences, that might be a better solution here. So we do show that for proteins which don't have any MSAs, our structure prediction tool does on an average somewhat better than AlphaFold type predictions. So there is another case where you should be mindful of using AlphaFold or any sort of machine learning tool. The idea is that ML systems without proper bioawareness could be critical. This is a LRR. This is a leucine-rich repeat protein, which could be used, which is important for a lot of anti-inflammatory responses in the body. If you want to sort of make a small peptide out of this protein, so if you want to piece out a small part of this protein, just take out a couple of beta strands, and if you want to predict the structure using AlphaFold of those couple of beta strands, if you feed that sequence to AlphaFold, AlphaFold is going to remember, hey, I saw this whole protein as something in my training set. I remembered that this is a part of this whole protein. It's going to give you a structure as if it's still a part of the big protein, even though there are no physical forces which could be rationally explained that can hold the beta sheet, couple of beta sheets like that. So that is something that one needs to be mindful of. So I have one last story. So currently an application of this that we are using, application of protein structure prediction and how to leverage protein design for meaningful applications. So this is called the computational tracking of escape variants of viruses. This is done by my student E. He just graduated yesterday. So generally the idea is that, so for during COVID, we've seen there was a lot of loss of jobs. People could not pay their rent on time. That's because the virus was quickly escaping. So there were several million deaths worldwide, and there were 700 million cases of infection. So that's primarily happening because the viral spike protein acts as an amazing biosensor. What it does is that the viral spike protein learns to quickly avoid the antibody that is trying to capture it and still retains high enough binding to the entry receptor. It just does it. Every mutation is capable of achieving two goals at the same time. So this is basically called viral escape. We tried to set up a tool that could actually predict future virus mutations by simulating this escape and see if we can recapitulate any of the. So with that, I'll sort of end, and this is my current group. We have some funding support from federal sources as well as some industrial sources, and the lab has been pretty new. We've been in the lab for like 1.5 years, so this is my second year in the university, and we've grown quite fast. And this was not possible without the students, and there's a lot of pretty talented students across the world that are part of my lab. So, with that, I end, and happy to take any questions or go into any part of the slides that you want to know a bit more about.
Thank you, Professor Chowdhury. Fantastic talk, I must say. A very interesting explanation of how AlphaFold actually works. I heard it for the first time. Thank you.
Yeah.
We are open to questions. Please feel free to feed in the chat your questions. Meanwhile, Dr. Roy or any of our company members have a question, they're welcome to ask. We have one question already. Hidaya, won't the protein structure change as the hydrophobic region is exposed to water and tryptophan will try to move away from water? That might generate an unstable structure, I think. This is a question and a comment from Hidaya.
Yeah. So for beta barrel proteins, beta barrels don't move away from in the presence of water, because the beta barrel structure is held together by hydrogen bonded contact with the backbone, not really with the side chain. The side chains do not really hold a beta barrel structure. So even if you prefer, so beta barrels are more stable for mutations than alpha helices. That's primarily the reason we chose a beta barrel protein.
Thank you. Next question is by Shubhendu. Are AlphaFold models generally better in terms of quality compared to those generated using classical homology modeling?
Oh yeah, that's for sure. So if you're using homology modeling, I'm assuming you have homologs, and if you have homologs, AlphaFold will be better. So the objective answer is that if you have homologs, so if you go to this picture, hold on, I have a picture over here which I showed. One second. Here. So if you look over here in this plot, so you can just take a look at this figure 5a of AlphaFold and sort of get an estimate of what to expect. So if you have less than 30 effective residues, there are ways to correct. If you just perform a p-blast, the number of sequences is not really your n effective. N effective is a more clustered set of sequences. So now you can imagine is that total number of truly unique sequences in your alignment, because alignments can give you exactly similar sequences as long as they're reported in literature. So if you can come up with 30 unique sequences from the family, then you can expect that the structure prediction would be much better for AlphaFold than any homology model.
Thank you. Next question is, what is the basic difference between Rosetta and IPRO design algorithm?
So the difference between Rosetta and iPro design approach is that, so with Rosetta you don't really make a mutation for a given task. What you do is Rosetta is a what's that called, random sequence generation tool. It uses a Monte Carlo approach. So essentially you would take a protein and mutate everything everywhere all at once, kind of. So you make based on how much compute power you have, you will generate all the protein sequences. So like if you perform point mutations, you are going to generate all the 20 alphabets and see which one actually works better. And then you are going to use that as a starting point, and then again generate all the possible variants that are there from that starting point. And you will the objective function. So if you want to improve binding to something or eliminate binding to something, that objective function is not a part of the design cycle. That's tested outside. You have to separately code for it, and then pass your sequences back in the for loop. But iPro is an integer optimization program. So if you have done simplex at any point during linear algebra courses, so this is that. So in this case, we qualify the protein structures beforehand for their interface. We describe the interface with the residue interaction energies, and it is a global optimization tool, so it does not have to screen through all the possible mutants. It knows how to perform, how to come up with changes that will give the best gain in terms of the objective function. So essentially, you can codify the objective function directly within the algorithm. And secondly, this is also deterministic. If you run iPro 20,000 times, you're always going to get the same answer. If you run PyRosetta 20,000 times, you will get 10,000 times roughly similar answer. And every time there is a chance that depending on what seed number your computer actually starts with, it might give you a different answer.
Thank you. Next question is from Professor Nilanjana. The question is about pore design. Can you predict or at least rank the flow and flux of the variants?
Yes. So we have transport measurements. We do. I didn't get a chance to show to that because I wanted to cover a few things. So we have so the paper, the pore designer paper, it's called pore designer itself. So that paper has transport measurements done using explicit all atom molecular dynamic simulations. We can if you're performing water separations water movement is easier to see. Ions are harder. So what you can do is that you can create two bulk groups of this thing of water, and you have a membrane patch. And the membrane, we can choose the membrane based on what is being done experimentally. The lipid vesicle that we use similar kind of material to make a membrane patch, and we graft our protein in it, and we can perform simulations to see how long does it take for water to go through, what is the residence time, what's the pore size distribution. Pores also breathe during the process of transport. So we can measure all of that, and then we can experimentally quantify using stop flow light scattering experiments.
Thank you. As of now, I'm taking few, almost all questions because we have some time.
Sure.
The next question is by Dr. Subbarao. Are there any success in modeling of membrane proteins, particularly GPCR?
GPCRs are hard. That's why I think GPCRs are hard largely because of the general uncertainty and the allostery and all of these. I would say there is nothing that can at the moment that can tell you what exactly. There's no one-stop solution for all membrane proteins. I think that's the better way to take machine learning tools. There is, I mean, this is also my take. I think this, again, take it with a pinch of salt if you have to, is that I don't think there can be any single protein structure prediction or protein design tool that works for anything that you want. You still have to retain your biological awareness, and you need to know what you're looking for. You always end up getting what you look for, but you always miss the things that you're not looking for. So similarly for GPCRs, if you want to model like a particular domain of a GPCR, I think you might be fine. But if you're saying that I want to model everything, any sort of interaction in the membrane bound form of GPCRs, I think that is still hard. There is nothing that can predict that yet, given the amount of allostery it shows.
Thank you. Next question is from Dr. Aishwarya. How accurately the binding pockets for ligands or small molecules predicted from ML models? Can we take these structures directly for performing drug discovery studies?
So if I understand the question properly, so this is Could you repeat the question, the first part of the
I'll
question?
repeat it. So, how accurately the binding pockets for ligands or small molecules are predicted from ML models? Can we take these structures directly for performing drug discovery studies?
So with my experience with drug discovery, again, it also is very context. Again, as I said, like these questions again need to be taken on a case by case basis in terms of what biological question are you asking. So if your drug discovery is for something like kinases, or which is for cancer, if you're looking at cancer-based drug discovery, then kinases have only one domain which is allosterically amenable. They have some sort of a glycine-based motif. I'm forgetting the motif. It's a three amino acid few sort of next to each other, which sort of open up like a gate. So you have to care about that. So the pockets are largely characterized. You can see a lot of literature which characterize the open and closed for conformations of those gates. Yes, I remember. It's a DFG motif. It's aspartate, phenylalanine, and glycine motif. But if you're looking at pain, so for example, if you're looking at pain, the proteins that are involved right now for drug binding are much larger. These are 1500 amino acid long single proteins, which are voltage gated channels. Now they're transporting ions at the same time. That's why they're opening and closing at the same time. They are bound by motor neuronal cell membranes. So you cannot just simulate it with like any other cell membrane. You have to make sure that the constitution is like a motor neuron. And also you are trying to see which way does the drug enter to bind to it. And there is ton of allostery. First of all, there is no structure prediction tool that can predict their structure right now. If you go to AlphaFold, plug in the sequence for basically a voltage gated ion channel, you will get like 70% loops. So the thing is, first of all, I think so this is very context dependent. There are problems which are very, very hard in this space. But if you're looking at drug binding to a very ordered protein, which is a small protein, roughly 300 amino acids long, structure largely solved, then I think you can rely on a ML-based tool to find its pockets. But again, it's not, these geometric optimization tools have been there since decades. It's not just a machine learning tool that is able to find it better than others. I think it's better to use a machine learning tool for using it as a discriminator, saying that is this binding good enough? Because let's say if you have two proteins, if you have a protein A and a protein B, and you have three drugs to test. So in the first protein, you want to see which one inhibits the proteins better. So if the first protein gets inhibited by like a KD value of something, let's say 10, it does not mean, and the second one gets inhibited by a KD value of, let's say, 100, it doesn't mean that the second one is necessarily worse. It could be that's the best that you could do with that protein, and that might be enough. So one needs to really understand experimental data before plugging in data or making inferences from machine learning models.
Thank you. I would request Smita to rephrase the question. Meanwhile, I'll go to the next one. Anil is asking, in your first part of the talk, what was the rationale for insertion mutations? Perhaps the deletion gaps were part of the evolution process itself.
So insertions and deletions are two sides of the same coin, right? So if you have a starting sequence and that sequence is shorter than any other family member, that means there is at least one position where you can insert an amino acid. So that's how you would find an insertion. But the tool does not necessarily restrict you to use the default option. So if you're clueless about where to make an insertion, then this would be the default approach in the tool. But if you want to actually make an insertion forcefully at a given point because you know something about it, the tool will let you do that.
Thank you. Next question is by Dr. Roy. How selective are the pores? Water purification for dialysis could be one application. Is that possible?
Yeah. The selectivity of the pores are good. I think there is a selectivity is, I forget the selectivity numbers. I think it's there reported in the paper. But the major issue with using these pores for water purification is that we've tried to, if you want to use like a big membrane, like so far we've been able to synthesize 1 centimeter square of a membrane, not like a full scale membrane, because these membranes are extremely brittle. So typical membranes, you have to sort of lift up from the aqueous phase and sort of roll it up very quickly. So there's only one company out there called Evonik, which has been able to perfect this technology of rapidly rolling regardless of what the membrane material is. But for our membranes, we still don't have a way to make it into a full scale membrane. It is still a lab scale membrane. So I'm not sure exactly how much of a surface area we need for performing dialysis, but there's some preliminary evidence that we are seeing that it can reject salt.
Thank you. Next question, how can we validate the AlphaFold structure if no reliable experimental structures are available? And how about iTaser for protein modeling?
Yeah. So I do show iTaser here. So iTaser is at the left bottom over here. So iTaser is essentially like a physics-based folding model. So you make a polypeptide chain using something like PyMol. It is just randomly adding amino acids after each other to form this like necklace-like structure and let it run for eternity. And it becomes, and if it folds in the right space, that is true. That is fine. The problem with iTaser is that it can get stuck in a local minima. So the local minima is that, so when a protein folds, from the back of a ribosome, you have to realize that the real process of biological protein folding needs to be always kept in mind. So the real process of biological protein folding happens instantaneously as the protein comes out from the back of a ribosome. So only one consecutive amino acids are getting produced by the process of translation, and they get folded. So the protein folding operates on consecutive amino acids right then. And if there are concerted changes that has to happen on amino acids that have already come out before, that will also happen right then. But it never starts folding after the whole protein has come out and now it starts folding. It does not happen like that. So with iTaser, that's the leap of faith. With iTaser, you have the full unfolded protein out there. Now you start folding it. Now when you start folding it, if so, let's say the middle of the protein starts folding first, it might be altogether going to a completely different trajectory that might end you in a different end state. So it is a lot about, you can do statistical thermodynamics on these things to figure out what is the probability that iTaser will actually get you to the right structure. But I think there is it also scales poorly with the length of the protein. So that's pretty much what it is. With AlphaFold, it does not really fold the protein. It's not solving the protein folding problem. It's already giving you a folded structure of a protein under a couple of constraints. Imagining that it was crystallized at 7.4 pH only. It has got no obligation to predict how the protein will fold at like a 5 pH or a 10 pH. So that's what it is doing. So just be mindful of when you're using what you're using.
Thank you. Actually, this points to another question by Dr. Subara. Do we need to do MD simulation AlphaFold or RGN2 models? So the question is not clear, but in case
Hey, no, I get the question.
Yeah.
No, I get the question. One definitely needs to do either at least some sort of energy minimization through Pi Rosetta or any other charm or anything. I mean, whatever force field is best for your work. Again, force fields also are different for different tasks. There's no one-stop solution for all. So depending on the problem you're looking at, I think you have to find out first the right force field that you want to care about. So then I think you should always minimize the structure to see if any bond strain gets relaxed at the end of, like, you can't take the structure predictions from static structure prediction methods like AlphaFold, ESM fold, Rosetta fold at face value. I think everybody actually sort of does that.
Thank you. I'll take the last question. Smita is asking, will AlphaFold model work for intrinsically disordered proteins?
Yeah, so that intrinsically disordered protein, that's a good question. I think this is actually a good question. So I will tell you something. Okay, so I think I'll need to use the drawing board for this. Give me 1 second. I will just insert a slide, and this is actually a good question. So so with intrinsically disordered proteins, the problem is they are disordered because there is very poor homology. So if you have this, you can still see my screen, right?
Yes.
Yeah. So okay, so this is the protein, and then this is the structure. Like this is the structure of the disordered protein. And if you are saying, so they have a lot of sequence variability. Okay, they have a lot of sequence variability, which is why they don't really have a fixed structure. So same with CDR3 loops of antibodies, if you know what I'm talking about. So if you make a mutation here, that mutation, so if I'm looking at the amino acid frequencies, so if I just plot a frequency of different amino acids seen in the family of the IDPs that I have, and this is the positions, frequency of alphabet, and this is the position, I am actually going to see like a very even distribution of a lot of different alphabets for IDPs. So which means that if you make a single point mutation, the sequence alignment actually does not change much. So this comes to AlphaFold as no real signal. It comes to it as a noise. So it's going to douse that noise out, and it's going to give you the same structure as that of an IDP that's in its training set. So it definitely has some IDPs in its training set. It's going to be fully biased based on whatever it has seen before. And it says that, okay, this is why protein design cannot work with AlphaFolds. If you have a protein, you make a small change, the multiple sequence alignment actually remains identical. So that's why for the wild type protein, if this is the structure, and if you make one change, the sequence alignment is still the same. AlphaFold gives the same statistics, it'll give you the exact same structure. So the same thing works with IDPs. Hopefully it's clear. Please follow up if you have any other question on this.
Professor Chowdhury, it's fantastic talk, means learned a lot, and you are really working on the cutting edge of the science, right? Means fascinating. I really loved it. I will be going through your talk again because there are a lot of things, a lot of data I could not capture. So I need to go through one by one, and maybe you will have a follow-up discussions whenever possible. And I really like to thank you from company that you took your time out. It's a morning, most probably early in the morning, and coming over and giving a talk to people who are interested in this field and working in this field. And so thanks a lot. And we would look forward to having you again and again in future.
No, this is my pleasure. I heard like it was a very active audience, which is great to always see, because I think I'm not a big fan of Zoom, but Zoom has facilitated this kind of stuff also. This is really helpful. And I think your audience was very amazing. I think they asked a lot of very good questions. So this has really been a great pleasure.
Thanks, Dr. Chowdhury. Thanks a lot.
Sounds good. Have a good
one. As well, I would like to thank all the audience coming and listening to your talk and asking fantastic questions, which basically enrich the content of the talk as well. And thanks a lot for joining. And we keep doing this every month. We have Fusion Talk at the end of the month, mostly last week of the month, and keep bringing speakers around the world and listening from them, about various subjects and cutting edge science, and mostly where science and technology they cross. And we are trying to build a small community out of this and see how we can grow. The talk will be available on YouTube, so I encourage all of you to share the talk with your friends and colleagues in the scientific fraternity. So thank you again, and I should not close it just not by saying thanks to Anupam and the team. Anupam, Meghna, and Amir, they basically worked hard to advertise the talk, getting the registrations, getting everything running, and coming up late in the evening and hosting the talk. Thanks a lot. Thank you very much for joining. Yeah. Thanks. We'll chat.
Thanks. Yeah. Thanks. Yeah. Bye.